My name is Noel-Marie Plonski, and I am a graduate student in Dr. Pankiewska's lab at Kent State University. I am currently using bioinformatic approaches and tools to study viral host interactions. The title of my talk today is RNA Editing Landscapes, a new model for biomarkers discovery in neurological disease. The next generation sequencing race has led to huge advances in sequencing technology. And as these sequencing technologies become faster, cheaper, and more readily available, there is a corresponding exponential increase in the amount of RNA sequencing datasets being stored in databases, including the MCBI SRA database, which is where we found our datasets. Because of these advances in technology, it has led to a new way to study biological mechanisms of complex human disease, the omics approach. This started with genomics and mapping of the human genome, followed by transcriptomics and even epigenomics and proteomics. A recent search of PubMed showed steadily increasing amounts of publications involving RNA sequencing since 2008. There was also a steady increase in the journal articles involving transcriptomics in particular. However, of these transcriptomic studies, only a few mention global RNA editing patterns as the main focus. Although there are many types of RNA editing, we focus on adenosine deaminase acting on RNA, or ADAR. ADAR deanimates adenosine to inosine, which later gets translated as guanine, resulting in an A to G transition. There are three ADAR loci, ADAR 1, 2, and 3. ADAR 1 and 2 are known to have catalytic activity, whereas ADAR 3 is thought to only have a regulatory role over the other two ADARs. These ADARs are expressed and edited in many tissues, including the brain, and has been studied for its role in many developmental processes, including brain development. All three ADARs have known isoforms. ADAR 2 has an isoform that also undergoes self-editing to make it more catalytically active. ADAR 1 has three known isoforms, one of which, ADAR 1P150, which is expressed upon innate interferon immune activation to the TLR3 pathway. It has been shown that activation of the TLR3 pathway by viral infections increases innate immune signaling and ultimately increased transcription of a number of genes, one of which is ADAR 1P150. This means viral infections can lead to changes in ADAR editing footprint, not only in the virus, affecting its infectability and lethality, as well as unintended consequences in the human transcriptome. ADARs have also shown to increase transcriptome diversity without having to increase the genome size. One way to think about this is to compare it to a chocolate chip cookie recipe. Although the recipe stays the same, you can change the ingredients in each batch that you make. This will create a slightly different taste or texture to the batch of cookies, but they are all still a chocolate chip cookie from the same recipe. Just like with ADAR editing, this process can make a mistake, or the environment can influence the editing patterns. Say you accidentally grab salt instead of sugar, or even forget the baking powder altogether. These will make inedible cookies, and when this happens to RNA editing, it can cause misfunctioning or non-existent proteins. For a biological example, we can look at one of the most commonly studied editing sites in the GRIA2 receptor, the QR site, where this site needs to be edited at nearly 100% to stop calcium signaling at a certain point during development. If this channel is left in the unedited isoform, then it can create an excitotoxic condition in the cells, leading to cell death. Another commonly studied gene with known editing sites is the serotonin receptor 2C. There are 5 editing sites and 3 amino acids that are independently edited of each other, and together in different patterns create 27 known differentially functioning isoforms. These editing sites are located in the G protein coupled region and have been shown to affect both serotonin signaling and overexcitability of the neuron through the sodium-potassium pump. Serotonin signaling has also been implicated in mood disorders, including depression and suicide, since the early 1970s. However, these are only 7 of the 152 known ADAR editing sites located in 98 genes that make up the excitome. A gene enrichment analysis of these genes shows the excitome are involved in pathways responsible for cell signaling, ion homeostasis, metabolic function, and transcriptional and translational processes. This leads us to our central hypothesis that others have also shared in recent publications: maternal immunoactivation, postnatal infections, a second viral infection in childhood, chronic inflammation, chronic stress, and a lifetime of viral infections can all lead to alterations to ADAR editing profiles affecting neuroplasticity. This can lead to a spectrum of neurological symptoms. depending on the time and location of the changes to ADAR editing landscape. These neurological symptoms include symptoms as obvious as microcephaly, but as silent as mood regulation, aggressive behavior, and memory formation and retrieval. Therefore, ADAR plays a central role in brain development and function, helping fine-tune the control of neurotransmitter synthesis and release, as well as ion homeostasis. When things like viral infections or other environmental influences change ADAR editing profiles, it can lead to devastating consequences that can vary from individual to individual. So what does this all mean? Our lab previously showed evidence of ADAR editing in Zika virus to add to the list of viruses with known ADAR editing footprints. Furthermore, last year, we published an article in Bioassays explaining our hypothesis and providing evidence from congenital Zika syndrome. We were able, using our computational tool, which I'll explain in a moment, to show that Zika virus not only increases ADAR 1p150 expression, but also significantly alters ADAR editing patterns in cells that are phenotypically similar to congenital Zika syndrome. Like I mentioned, the first step was to create a fully automated pipeline using a new mathematical model to map ADAR editing profiles. We call this pipeline Automated Isoform Diversity Detector, or AID, which uses standard open source tools for genomic alignment, transcriptome assembly, variant calling, and variant prediction, combined with our custom EX toolset to create the ADAR editing landscapes. The new model includes item response theory utilizing Gutman scale and machine learning random forest analysis to score, rank, and order editing sites in any condition. When we talk about the rank order, we categorize into three categories. Category 1, edited in all samples. Category 2, edited in only some of the samples. And Category 3, not edited at all in any of the samples. To test one of the parts of our central hypothesis that changes to ADAR editing landscapes can be used to predict biomarkers for depression or suicide, we use publicly available data sets from the SRA database. The data sets together consist of 324 samples with 141 females, 183 males, of which 77 died of natural causes, 226 died of suicide, and the 21 accidental deaths were filtered out from our analysis. These samples also came from 6 different brain regions, all with implications in the biological mechanisms behind depression and suicide. These include the submiculum, important in working memory and drug addiction, the nucleus accumbens, which is involved in motivation and reward, reinforcement of learning, the anterior insula, linked to emotion, to body's homeostasis, empathy, perception, and other things. The cingulate gyrus, which is linked with mood and anxiety and memory, and self-esteem and processing of sadness. The dorsal prefrontal cortex, which is involved in working memory, cognitive flexibility, and planning. And finally, the orbitofrontal cortex, which is involved in emotion and reward, and in courting contingencies in a flexible manner. To confirm the variants we are looking at can be attributed to ADAR editing alone, we use a series of filters to filter out false positives by using quality control measures from alignment. Before filtering, AID predicted just over 80,000 variants. These variants then went through a quality control filter based on sequencing error probability. A second round of variant calling was performed, and only the sites that were predicted and not filtered during both rounds were kept as true editing events. But we still weren't done filtering. We found almost 30,000 of these variants were found in the SMP database, and once they were filtered out, we see only a few hundred that are not A to G, except a little blip at the C to T editing sites, which may be attributed to the other RNA editing protein ApoBac, which we will not talk about in this presentation, and these have been filtered out from our analysis. When we start looking at the global editing level, considering all ADAR editing sites transcriptome-wide, the only significant difference we see is between regions and between biological sex, but not between suicide and natural death. Natural death shown in blue and suicide shown in red. Continuing to look at the global level, we also look at the location of editing sites along the gene loci. Although we found no significant difference between suicide and natural causes, we did see about 80% of the editing sites are located in the intronic region, and less than 10% were found in the exon region. But there were no differences, again, between regions, biological sexes, or condition, cause of death. This is when we start looking at just the excitome genes mentioned earlier that have no consequences on protein structure and function. These were classified as either edited or not edited, and then the samples were assigned a score based on the Gutman scale. The higher the Gutman score, the more editing is present in the excitome. Here we see significant differences between suicide and natural causes of death. We see this in the biological sexes, the brain regions, and the cause of death. Here and for the rest of the presentation, remember that natural causes of death are in blue and suicides are in red. Here we also see the females on the top panel and the males on the bottom. And from left to right, you have brain regions divided in the panels. In females, editing is higher in suicide in the cingulate gyrus and lower in suicide in the prefrontal cortex. Females also show more editing in general and more significant differences in editing amounts. When we look at the Guttman rank order, we set 3 categories. Category 1, completely edited. Category 2, only in some samples. Category 3, edited in any sample, not edited in any sample. We set the Guttman order for the female anterior insula as a reference. Here the females are on the right and the males are on the left, and regions are from top to bottom. We see here that natural death, the editing patterns are pretty similar in females with only a few category switches. But when we compare the red line to the blue line, or suicide to natural causes of death, we see a significant number of changes between categories up and down. Males, on the other hand, show more differences in the blue lines, suggesting major differences between regions, but not necessarily between conditions. There is much less differences between suicide and natural causes of death in males, suggesting less of a role of editing in suicide in males than in females. These editing profiles can be used as a signature for each brain region, biological sex, and/or cause of death. But to predict the actual list of biomarkers, we use machine learning random forest models to find top differentially edited sites. Using these models, we created a model with an 18% error rate using excitome editing frequencies. These graphs here show the top editing sites and the prediction of suicide or natural causes of death. The more accurate the editing site is at predicting the condition, the further apart the grouping of the colors are. For example, if you look at the top row, these are the top 8 differentially edited genes, and we can see clear clustering of natural causes of death near the top and suicides near the bottom. And ranging from left to right, we're looking at percentage of editing at that site. Another way to look at this model is to use a scatter plot showing the 2 measures of accuracy when testing the model. And then ones shown down here in this corner are the most accurate. And the closer they are to the line, the more accurate they were on both measures. And we find similar top genes. If we take a closer look at the significant differences for some of the top regulated genes, we look at the serotonin receptor, again, only found in the nucleus accumbens. And we look at all 5 editing sites, which show differential editing, and they appear to be differentially edited in both males and females. Females showing more editing overall. This would suggest serotonergic tone and dopaminergic reward regions of the brain may be an underlying mechanism of suicide. We also see a significant difference in calcium channels, including the glutamate channels 3A2 and GRIC2 at multiple sites. Here again, we see differences between males and females, as well as brain regions and between suicide and natural causes of death. These would suggest a role for glutaminergic signaling and multiple regions of the brain underpinning suicide. However, these genes are not widely expressed anywhere but in the brain. So we may need to look at other genes that are expressed more frequently in tissues such as blood to be more clinically relevant. The top ranked genes, ACP1, which is the gene involved in cholesterol metabolism, shows significant differences in all brain regions and is pretty similar in editing patterns between males and females and in most regions. In almost all cases, suicide shows less editing than the natural cause. This is supported by studies that show the involvement of cholesterol metabolism in suicidal behavior. Moreover, this gene is also expressed and edited in blood samples, so it can be used as a clinically relevant biomarker. We will come back to this when I talk about future directions in a moment. Another gene that is also expressed in the blood as well as the brain is SRP9, known to play a role in the translation of proteins in endoplasmic reticulum in cells, and is high on the top of the editing site list. However, the editing frequencies are quite low and the differences between conditions are minimal. This would suggest it may be not such a useful marker in the clinical setting. The last gene I will mention from the biomarker list is IGFBP7, which is expressed in the brain and the blood and shows clear editing differences between suicide and natural causes. IGFBP7 is an insulin growth factor binding protein and has been shown to be more susceptible to proteolytic degradation with decreases in editing, like that seen in the subiculum, in the prefrontal cortex of females, and the cingulate gyrus of males. Changes to the concentrations of this protein present in the cell can cause changes to vasculogenesis via interaction with vascular endothelial growth factor. These potential biomarkers alone, or in conjunction with others and in combination with landscapes generated by AID, can be used as a way to profile risk of suicidal behavior, but most still needs to be experimentally validated. In summary, AID editing profiles of the excitome can be used to not only find biomarkers to predict suicidal risk, but also provide insight into the biological mechanisms, including alterations to calcium channel signaling and plasticity, dopaminergic reward pathways through serotonergic signaling, changes in the nucleus accumbens, vasculogenesis leading to apoptosis and decreased proliferation, especially in the PFC, OFC, and subiculum, protein translation in the endoplasmic reticulum leading to changes in plasticity, and cholesterol metabolism, which has been linked directly to impulsive behavior and suicidal ideation and attempts. ADAR editing profiles can also be used in other neurological diseases and disorders, and potentially with blood sample data sets instead of brain tissue. We are currently working on a publicly available data set with bipolar to find clinically relevant biomarkers, with the ultimate goal being a better diagnostic and prognostic tool for mental health care professionals in the treatment of mood disorders. For example, assessing an individual's response to drug therapy or risk for suicidal or homicidal behavior. However, much more investigation is needed and more data needs to be analyzed before this can actually be achieved. We can also use this to generate new hypotheses to experimentally verified. One of these is a stem cell project I will be starting soon, where we will be treating neural stem cells with a mimic to viral infections and then monitoring editing profiles and changes to calcium signaling in the cells as they differentiate to dopaminergic neurons to test the involvement of editing in serotonergic and dopaminergic cells of the VTA. We also plan on extending AID to create single-cell editing profiles in an attempt to better understand the spatial temporal regulation of ADAR editing on an individual cell basis, which is especially important in the study of brain function. With that, I'd like to thank the organizers for allowing me to talk, all the members of Dr. Helen Kankiska's lab, past and present, and some other managers listed here at Kent State, and other collaborators.